Stimulated Raman scattering microscopy is a chemical imaging technology that enables the rapid and quantitative detection of lipids and allows the tracking of lipids in live animals in a label-free manner.
The most important advantage of SRS microscopy over traditional lipid detection techniques is that it is label-free and therefore is not as sustainable to photobleaching as other fluorescence and staining methods.
The use of SRS microscopy, in conjunction with the genetic and biochemical tools available for model organisms, particularly Caenorhabditis elegans, provides a powerful framework for studying novel regulators of lipid biology and metabolism.
To feed laser beams into the SRS microscope, first set the periscope to lift the beam from a picosecond light source exit to the infrared laser input port on the scanner of the microscope, and open the laser control software to start the lasers. Lower the pump laser power to 50 milliwatts and set the pump signal to 750 nanometers. Use a beam expander to adjust the beam diameter to fit the back aperture of the microscope objectives. Use two relay mirrors, M1 and M2, to guide the laser beam to the periscope, and set the knobs of the periscope at the center of the tuning range. Select the initial position and angle of each mirror such that the laser beam approximately hits the center of the mirror to launch the beam into the microscope, and use an empty port on the objective turret of the microscope to perform the coarse alignment. Place the power meter.